Amino acids are building blocks of protein. We know that the structure of biological molecules determines their function. Amino acids have different structures, properties, and ionizing abilities. Therefore, the arrangement of amino acids in proteins will affect the structure and function of proteins. Amino acids can have an L isomer or a D isomer. L stands for levorotary, D stands for dextrorotary. Only L amino acids are found in proteins. There are a series of structures from primary through to quaternary. The primary structure is the amino acid sequence. The secondary structure is local spatial arrangement of the amino acids in the peptide chain, for example, a beta-pleated sheet or an alpha helix, both of which can be found in proteins. The organization of the primary and secondary structures into a 3D protein shape forms the tertiary structure. The arrangement of different subunits in a protein forms the quaternary structure. Amino acid sequences are defined by genes. The unique properties of proteins are determined by the order of the amino acids in their R groups. The order of amino acids is called the primary sequence or primary structure. Amino acid polymers. Many amino acids joined by peptide bonds are called a polypeptide chain or protein. Most natural proteins are made up of 50 to 2,000 amino acid residues. The amino acid chain is called the backbone. An amino acid in a polypeptide chain is called a residue. The different R groups are called the side chains. A polypeptide chain has direction because amino acids have different ends. The amino or N terminus is where the NH2 group can be found. The carboxyl or C terminus is where the COOH group can be found. By convention, the amino end, NH2 group of the chain, is taken to be the beginning and written on the left. The peptide bond. Amino acids are linked to peptide bonds to form proteins. This reaction occurs on the ribosome. A condensation reaction forms a covalent peptide bond between the COOH group and the NH3 group, forming a CONH. Peptide bonds have partial double bond characteristics. The peptide bond resonates between two forms, a double bond form and a single bond form. In the double bond form, the oxygen has donated a pair of electrons, so it's negatively charged. And in the single bond form, there's a double bond between the carbon and the oxygen. The main chain forms the inner part of the rod. The side chains extend outwards. This is the case in the alpha helix. In the alpha helix, the main chain forms the inner part of the rod. The side chain extends outwards. Hydrogen bonds between the NH and CO groups of the main chain stabilize the helix. These are intrachain hydrogen bonds. Each CO oxygen is hydrogen bonded to the NH of the amino acid residue 4 residues ahead. In the linear sequence, you're gay. Each CO oxygen is hydrogen bonded to the NH of the amino acid 4 residues ahead in the linear sequence. Rod-like structures. It's a rod-like structure. Hydrogen bonds are nearly parallel to the helix axis, and there is some elasticity in the helix. The secondary structure, 2, the beta-pleated sheet. Stabilized by hydrogen bonding between adjacent strands. It may be between two parts of the same chain, intrachain, or between different amino acid chains, interchain. Fully extended polypeptide chain. No elasticity, zigzag pleated shape. R groups lie outside the plane of the sheet. So it's a fully extended polypeptide chain. There's no elasticity. There's a zigzag, pleated shape, and R groups lie outside the plane of the sheet. Many proteins contain both alpha helices and beta sheets. Tertiary structure. Proteins have compact three-dimensional structures, but they are flexible molecules. Tertiary structure is the overall 3D shape of protein subunits. It involves the close packing of secondary structural elements. The interior of protein is hydrophobic, i.e. contains leucine, valine, met, or Phe, etc. The exterior of proteins is mostly hydrophilic, i.e. contains charged residues, arginine, histine, lysine, asparagine, and glutamine, although most proteins do have small exposed hydrophobic regions. The tertiary structure is important for positioning of binding sites and active sites, etc. The tertiary structure, the active site of an enzyme. Quaternary structure. Some proteins contain more than one polypeptide chain. The individual polypeptide chains are called subunits. Quaternary structure is the arrangement in space of the subunits. Quaternary structure. Although many proteins function as monomers, e.g. myoglobin and insulin, multi-subunit proteins are very common in biological systems. For example, structural proteins, collagen monomers join together to form collagen fibers, multi-enzyme complexes, such as mitochondrial ATPase, immune system proteins, such as antibodies, transport proteins, such as hemoglobin, motility proteins, such as myosin. The interfaces between subunits contains closely packed non-polar side chains, hydrogen bonds, and sometimes disulfide bonds. The interfaces between subunits contain closely packed non-polar side chains, hydrogen bonds, and sometimes disulfide bonds. Interactions between subunits can influence the structure and therefore function of the neighboring subunits. The subunits can often transmit conformational changes through the complex. Structural subunits maintain appropriate conformations of neighbors and increase stability. Genetically economic, as they have less DNA code needed. The speed of the reaction is effective, as the active sites are close together. It has cooperative effects, so there's communication effectively. There are interactions between subunits which can influence the structure and therefore function of the neighboring subunits. The subunits can often transmit conformational changes through the complex. Forces that stabilize tertiary and quaternary structure. The packing of helices, sheets, and different subunits in proteins is stabilized by side chain interactions. Electrostatic interactions, hydrogen bonds, van der Waals forces, burial of hydrophobic residues, and disulfide bonds. Electrostatic interactions stabilize tertiary and quaternary interaction by electrostatic interactions between oppositely charged side chains. This is called an ion pair. The interaction is called a salt bridge. Attraction between an hydrogen atom of a donor group, e.g. OH, and NH, and non-bonding electrons on an acceptor group, e.g. OC, stabilizes tertiary and quaternary interactions by hydrogen bonds between the side chains. Aspartic acid is an acceptor and a donor. Serine is a donor. Most polar residues can hydrogen bond 12 of the amino acids. Van der Waals interactions in electrically neutral molecules. Electron density around atoms is uneven, creating regions of slightly positive and slightly negative charges. These are dipoles. They can induce an opposite charge in an adjacent atom. Non-covalent forces of attraction between side chains with different dipoles. All amino acids form these. The hydrophobic effect. This is most important effect in stabilizing proteins. Non-polar amino acids try to minimize contact with water and are buried in the core of the protein in aqueous solution. Polar amino acids are found on the surface of the protein or are hydrogen bonded to the core. Disulfide bonds. Oxidation between pairs of cis and adjacent in you're gay. Disulfide bonds. These can be as a result of oxidation between pairs of cis, and they are adjacent in space, not sequence. Protein structure is important. Proteins need to be correctly folded to be functional. How do proteins fold? A peptide, 100 residues long, would take 20 billion years to randomly try all the possible conformations available to it. But folding takes blah 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 blah. Small sequences of secondary structure form. Stabilizing forces tend to retain these. Less stable structures unravel, and alternative structures form in quick succession until a more stable conformation occurs. Folding continues in a hierarchical manner. Tertiary and quaternary structures form subsequently. What happens if proteins fold incorrectly? Cellular process relying on the presence of the correct protein will not continue, for example in phenylketonuria. The misfolded protein may accumulate and hinder normal cellular processes, for example Alzheimer's disease. The misfolded protein may cause conformational changes in other proteins, e.g. with Creutzfeldt-Jakob disease. The cell may identify the peptide for early destruction,